Good morning. So I'd like to start by thanking Carlos for that very nice introduction and the organizers, especially David, for this invitation to speak at this very special event. I wish I could be there at Caltech in person, but it's great to have this opportunity to share some of our work with you. So as Carlos mentioned, our lab uses the Songbird as a model system to understand how the brain generates complex learned sequential behaviors, and to understand the brain circuits that do this at a detailed, mechanistic, quantitative engineering level of description. So young songbirds learn their songs by imitating their parents, and they do this in a series of stages. At an early stage, they listen to their parents sing, and they form a memory of that tutor song. Then they begin babbling, and over the course of several weeks, their song develops repeatable structure until a few weeks later, their song converges onto what can be a pretty good copy of the parent's song. Now, the basic framework for how we think this happens is reinforcement learning, which was first introduced to the songbird field by Kenji Doya and Terry Sinowski in the late 80s. So the basic idea is that young birds have a neural circuit for producing song, as well as a source of variability so that they can try out different variations of the song. Then by listening to themselves, they compare the produced songs to the auditory memory of the tutor song. And that comparison results in an error signal or a reinforcement signal that's used to modify the song motor circuitry. And the idea is that this results in the motor circuit reinforcing good song variations and suppressing bad variations. And that eventually leads to the good imitation. So what I'd like to do today is to describe a connectomic approach to testing a specific circuit hypothesis for how reinforcement learning is implemented in cortical basal ganglia circuits of the avian brain. I'll describe a preliminary model and describe some of the ultrastructural predictions then for how motor and timing signals are integrated at the synaptic level to implement a hypothesized learning rule that's important in that model. And finally, I'll describe a collaboration with Winfried Denk at the Max Planck Institute and Viren Jain and his team at Google to test those predictions in dense EM level reconstructions of the songbird basal ganglia. So the basic brain pathways involved in producing and learning the song are fairly well known. There's a motor pathway that generates a sequence of motor commands that control the vocal organ. At the top of this pathway is a brain area HVC, which controls the timing of the song. Neurons in HVC generate a single burst of spikes at one moment in the song, but as a population, they represent all times in the song. Now, there's also a separate pathway that's involved primarily in learning and whose main function, first proposed by Sarah Bottcher, is to program up the motor pathway to produce the correct sequence of motor gestures. And this circuit was shown by David Perkel, Alison Dope, Eric Jarvis, and many others to comprise a cortical basal ganglia thalamocortical loop, similar to those found in mammals that underlie motor learning and, as Anne just described, habit formation, among other things. Now, one component of this loop is a cortical circuit LMAN that's thought to essentially be a variability generator. Lesions of this area eliminate the variability inherent in juvenile birdsong. In gray, you can see the large pitch fluctuations as a function of time during one syllable in a young bird with intact LMAN. In contrast, in red, you can see the much reduced pitch fluctuations in a young bird that's had LMAN inactivated. Now, the basal ganglia component of this pathway is area X. It has mixed striatal and cell types, and even has cell types associated with the subthalamic nucleus. And it's been proposed to carry out a very specific function, and that is to determine which LMAN variations made the song better and which L-man variations made the song worse, and then use that information to bias the variations in L-man, the activity in L-man, to produce the good variations more often. So how does Area X figure out which L-man variations lead to a better song and which lead to a worse song? So every axon that goes from L-man to the motor pathway sends a collateral to Area X. So Area X sees an image of what every one of the 6,000 L-man neurons is doing at every moment in the song. So now Area X also receives a dopaminergic input from the ventral tegmental area that in turn receives a projection from a descending auditory pathway. And our lab and others have shown that this circuit generates at several stages of this pathway error-related signals during singing. Now, the best evidence that this pathway actually can serve as a reinforcement signal for vocal learning comes from a beautiful study from Jesse Goldberg's lab. Vikram Gadekar, a postdoc in Jesse's lab, used antidromic identification to find and record VTA neurons that project to Area X. They then played noise bursts targeted to a particular syllable during the song of a bird while it's singing. But they played that noise burst on a random 50% of song renditions to trick the bird into thinking it made a mistake on those renditions. So on those, what they found was that on those trials where the noise burst was played, the VTA neurons exhibited a brief suppression of spiking. And on those trials where the noise burst was not played, the VTA neurons showed a brief excess burst of spikes. So this can be thought of as a type of performance prediction error signal, much like the kind of reward prediction error signals described by Wolfram Schultz and others in the mammalian ventral tegmental area. And in those studies, you may recall, VTA neurons exhibited a burst of spikes in response to unpredicted rewards and a suppression of spikes when a predicted reward was absent. So in principle, now with this VTA reinforcement signal, Area X could now correlate the L-man activity with song performance and figure out which variations make the song better. Now, it's important to note that this isn't really enough to implement song learning. And the reason is that a particular L-man fluctuation, for example, that leads to added muscular contraction, might make the song better during one song syllable, but that same fluctuation could make the song worse in a different syllable. So to solve this problem, the correlation of L-man activity with song performance really needs to be done independently at every time in the song. So there's also a projection to Area X from this timing circuit HVC, and that carries the same kind of sparse code for timing that it carries in the motor pathway. So with this information now, with this timing information, Area X can figure out which L-man fluctuations at each time in the song make the song better at that time, okay? So of course now, once Area X has figured that out, Area X can in principle bias L-man activity through this thalamocortical loop to produce good variations more often and the bad variations less often. Now, because L-man is then in turn biasing the motor pathway to produce patterns of activity that lead to a better song, those good patterns can be actually kind of implemented into the motor circuit by a simple Hebbian learning rule. Okay, now we've also proposed a detailed circuit implementation of this process in Area X. And the idea is that L-man inputs and HVC inputs converge on medium spiny neurons of Area X, where they implement a specific learning rule. And that is that coincident activation of L-man input, HVC input, and VTA input leads to a strengthening of precisely those HVC synapses at which the coincidence occurs. And notice that this is not just classical Hebbian learning that's driven by paired pre-post activation. This is a different learning rule that requires 3 different factors. And that learning rule is similar to other ideas that have been proposed, for example, synaptic tag and capture, and 3-factor learning rules that have been proposed by others. And it was also very much inspired by the work from Michale Fee and Sebastian Sung on the idea of node perturbation as a mechanism of gradient descent learning in neural networks. So here's how this works. As the bird sings, HVC neurons are sparsely active at each time. If at one particular time point, let's say this time point 3 here, the L-man neuron is active, and that leads to a better song outcome, namely resulting in activation of VTA, this would lead to the strengthening of this particular HVC to MSN synapse. Now, the next time the bird sings, that strengthened synapse would allow that HVC input at time point 3 to activate the MSN, which then through that thalamocortical feedback pathway through the pallidum would reactivate L-man at that time point, leading to a reliably better song performance at that time. And of course, that learning is happening independently for all the different L-man neurons at all the different time points, and that would lead to a global improvement of song performance. And you can see that this kind of learning rule leads to a temporally dependent bias signal that would then drive the appropriate patterns of activity in L-man at each time. Now we've implemented this, this model in a simple computational model. And one of the things that we found is that one of the cool things about this model is that it provides this circuit with a temporal resolution that's actually much faster than the time course of the dopaminergic signal. So we've confirmed in this model that the temporal resolution of the model is determined not by the duration of the dopamine signal, but rather by the coincidence of the L-man and HVC inputs, each of which have a temporal resolution in the bird of only 10 milliseconds. And so you can see that the impulse response of that bias signal, in this case, can be only 10 milliseconds, which is much shorter than the 100 milliseconds or so dopaminergic time course. And that agrees with results from Michael Brainerd's lab that have shown that songbirds actually have very short, fast temporal resolution in their learning. So by targeting a particular part of a syllable with a noise burst, you can get learning that occurs in a 10 millisecond or so window. Now, I want to point out that in this model, there's a striking asymmetry in the role of L-man and HVC inputs. So HVC synapses, you can see, convey timing information, whereas L-man inputs convey a collateral discharge or an efference copy of action signals. HVC inputs play a role in driving the MSNs to produce this bias of L-man activity, whereas L-man inputs serve to gate plasticity, but not activate the MSNs. In this model, the HVC synapses are the plastic synapses, and L-man synapses are not plastic. And finally, the plasticity at HVC synapses has to be highly selective. In other words, you need to be able to drive plasticity in one of these synapses that's active at one time point, but not neighboring synapses that are active at different time points. In contrast, the L-man input is a global signal that has to gate plasticity at all of the HVC synapses on an MSN. Okay, so we have hypothesized that this proposed learning rule and the distinct properties of these different inputs could be implemented by a particular structural arrangement of synapses onto MSN dendrites. And that is having HVC inputs terminate on dendritic spines and L-man inputs terminate on dendritic shafts. Thus, L-man inactivation would depolarize, for example, an entire dendritic shaft, removing magnesium block from NMDA receptors at all the spines on that dendrite. Coincident HVC input would then lead to calcium influx, which could form a biochemical eligibility trace, which then remembers which actions happened at which times. Subsequent arrival of a dopaminergic input would lead to synaptic strengthening of precisely those synapses that need to be changed in this model. Okay, so how do you go about testing this model? So there are many ways you could imagine going about doing this electrophysiologically, using optogenetics. Today, I want to describe to you a particular approach that we've taken to test this model. This is work done with Jürgen Kornfeld and our postdoc in our lab in collaboration with Winfried Denk at the Max Planck Institute and Viren Jain and his team at Google, in which we set out to test various aspects of this model, starting with the structural asymmetry of HVC and LMAN inputs onto MSNs. So Jürgen, when he was a graduate student in Winfried's lab, carried out serial block face electron microscopic imaging of two samples of area X tissue. One was a cube about 100 microns on a side, and the other one was roughly 300 microns on a side. Now, as you know, in this method, one takes an EM image of the surface of a block of EM stained and embedded tissue, and then shaves about 10 nanometers off the surface, images again, slices, images, and so on. And this produces a stack of tens of thousands of serial EM images through the sample. Those images are then all aligned to each other carefully, which is relatively easy in this method because all the images were taken off of a fairly stable block of embedded tissue. So, okay, so Viren and his group then used their new flood filling network algorithm to carry out a dense reconstruction of all the neurites in the volume and agglomerate those into complete neurons or neuron fragments. Jürgen then used a neural network-based classifier that he developed to identify the synapses in that volume and characterize them as excitatory or inhibitory based on established criteria. He used another neural network classifier that he developed to identify neuron types based on morphological features. And again, the ground truth on this was established from criteria in the mammalian and birdsong literature on striatal and basal ganglia tissue. So here you can see the automatic cell type classification shown in different colors, overlaid on an independent UMAP representation of the different neurites using combined neurite morphology and connectivity. And you can see that there's a fairly clear distinction of cell type identification among these different clusters. On the right, you can see a single reconstructed medium spiny neuron. Here is a reconstructed LMAN axon and a reconstructed HVC axon. Now these axons could be, these LMAN and HVC axons could be identified fairly easily in the volume because we know from single cell fills and light microscopy that individual HVC axons branch rarely and arborize throughout the volume of area X, the entire 1.5 millimeter extent of area X, while LMAN axons arborize in a tight terminal plexus that's about 100 microns across and that occupies about 1/1500th the volume of area X. So what about the synaptic contacts of LMAN and HVC axons onto MSNs? So here you can see an example of an MSN dendrite with a synaptic input onto a spine head from an HVC axon and an input, synaptic contact from an LMAN axon onto the dendritic shaft. We carried out a detailed quantification of individual axons of the HVC and LMAN axons of the fraction of the presynaptic contact area of that axon that forms synapses onto MSN spine heads. So this is the fraction of presynaptic contact area of the axon that contacts spine heads. And what you can see is, what I'm showing here is the distribution of that spine head fraction across all of the HVC axons and across all the LMN axons. And you can see that most HVC axons have more than 80% of their synaptic contact area onto MSN spine heads, while the majority of LMN axons have a much smaller fraction of their synaptic contact area onto spines. Now, I'm not showing the data here, but we've also found that the few spines that do receive LMN inputs are significantly smaller than those that receive HVC inputs. And at the bottom here, you can see a simple graphical summary of the way HVC axons and LMN axons distribute their connections onto spines and shafts. And you can see that LMN axons have a preference for making contacts onto dendritic shafts. Okay, so we have clear evidence then of a structural asymmetry in the way HVC and LMN axons contact MSN dendrites. Again, HVC axons prefer to contact dendritic spines, LMN axons prefer to contact dendritic shafts. Now, there's another aspect of our model that we've begun testing. Now, it's clear from this model that LMN inputs need to depolarize MSN dendrites to gate learning, but they can't allow the MSN neuron to spike because this would inappropriately reinforce LMN activity in a way that's independent of the learning process. So we've proposed that there may exist a selective feed-forward inhibitory pathway, potentially involving striatal fast spiking inhibitory neurons, the function of which would be to allow LMN axons to depolarize the dendrites of MSNs, but suppress the actual spiking of the MSN soma that would normally result from that depolarization. So Jürgen has dug into reconstructed neurons in the larger data set and found that it contains many examples of known striatal and palatal neuron types. By far, the most numerous cell type in area X is the MSNs, which are shown in this cluster, downsampled by a factor of 20. But you can see in the larger data set, we also see clear clusters of many other neuron types, including GPE and GPI palatal cell types, low threshold spiking interneurons, that's a striatal cell type, neurogliaform cells, another strial interneuron cell type, even subthalamic nucleus, excitatory neurons that were identified by David Perkel in area X. Here is a cluster of putative cholinergic tonically active neurons, and here is our cluster of putative fast spiking neurons. So Jürgen began analyzing those fast spiking neurons, and what he found is that, of course, because this is how they were clustered, those neurons are very similar in morphology to labeled fast spiking neurons in area X that David Perkel identified using electrophysiological recordings. And those are easy to find because they have very narrow spike waveforms. Now, what about the source of excitatory inputs onto these putative fast spiking neurons? Well, Jürgen found that the distribution of synaptic contact area onto these fast spikers from LMN axons is actually about an order of magnitude larger than the distribution of synaptic contact areas from HVC axons. And this suggests that LMN is really the predominant input to putative fast spiking neurons in this circuit. Now, notably, that asymmetry does not exist for LMN and HVC inputs onto the neurogliaform cells, which are, as I mentioned, another local inhibitory interneuron in the striatum. Okay, so another really striking aspect of the population of fast spiking interneurons is that there's close to the same number of fast spiking neurons as there are LMN neurons, and therefore LMN axons. In other words, there's roughly a 1-to-1 correspondence between the number of FS neurons and LMN arborizations, L-man neurons in area X. In addition to this, the size of the dendritic arborization, which you can see in this reconstructed fast spiking neuron here in blue, closely matches the size of the L-man axon arborization, which you can see in orange behind. So this raises the possibility, the really interesting possibility, that fast spiking neurons could essentially serve as a private inhibitory channel for one or two L-man axons. Now, consistent with this, a direct analysis of L-man to FS connectivity shows that the vast majority of L-man axons, of the most completely reconstructed L-man axons, form synaptic contacts with only one or two FS interneurons, and very few L-man axons form synaptic connections onto more than two FS interneurons. Further evidence for that degree of selectivity comes from electrophysiological recordings done in our lab by Jesse Goldberg when he was a postdoc in the lab. These recordings show that FS neurons exhibit spiking patterns of random bursts that are remarkably similar to the spiking patterns of individual single L-man neurons during singing, again suggesting a low degree of convergence from L-man axons onto FS neurons. And finally, Jürgen analyzed the spatial distribution of L-man and FS synapses onto onto MSNs, and he found that the feed-forward inhibitory synapses from FS neurons are significantly biased toward the soma compared to inputs from the excitatory L-man axons, which avoid the soma and preferentially target dendrites, as I have been hinting at in this circuit diagram. So altogether, we think these findings indicate that there might exist a highly focal channel of feed-forward inhibition from each L-man axon to the MSNs it innervates. And this pattern of excitation and feed-forward inhibition would allow excitatory inputs to depolarize the dendrites to gate plasticity during the burst of an L-man neuron, while not allowing that input to interfere with the actual spiking of the MSNs during that input. Okay, so let me end by summarizing. The connectomic data that we've been analyzing in collaboration with Winfried and Varen have allowed us to test a specific hypothesis for how reinforcement learning is implemented in a cortical basal ganglia circuit. It's allowed us to look at how different signals important for that learning process interact at different subcellular compartments, in particular to implement this hypothesized three-factor learning rule. And in the process of that, we've also identified a feed-forward inhibitory pathway whose function may be to allow excitatory inputs to depolarize dendrites to gate learning, while suppressing the spiking in those target neurons, which would interfere with the ongoing computation that those neurons are doing. And we're very excited about this, the possibility that this may represent a general principle in learning in striatal and basal ganglia circuits. So I'd like to end with special thanks to our collaborators, Winfried, Varen, and Michale. I also want to give many, many thanks to Jürgen Kornfeld in our lab, who carried out all of the data acquisition and analysis in the connectomic work I described. He also developed many of the analysis techniques that were used in the process of that. So thank you very much for your attention.